This is a special episode of the Stem Cell Podcast, ISSCR 2026 On the Ground. Hello, everybody. This is Dr. Daylon James here with Dr. Arun Sharma. Welcome back to the Stem Cell Podcast, where we culture knowledge in stem cell research by talking to some of the brightest minds in the field. The podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. Today, we're bringing you a special episode recorded on site at the ISSCR 2026 annual meeting in Montreal, Canada. Held July 8th to the 11th, the meeting brought together stem cell researchers and scientists from around the world for several days of exciting science, new discoveries, and great conversations. While we were there, we had the chance to connect with delegates from across the global research community and hear about their perspectives on the meeting, the science that stood out to them, and the future of the field. So today, we're sharing some of those conversations with you, truly talking to some of the brightest minds in the field. But before we jump into things,
do you work with human pluripotent stem cells and want to strengthen reproducibility in your research? Learn how with applying the ISSCR Standards for Research, a free on-demand training course offered by STEMCELL Technologies and the ISSCR. This self-paced online course provides practical guidance on best practices for cell characterization, pluripotency assessment, genomic integrity testing, and reporting. To learn more and register, visit stemcell.com/ISSCR-standards-course. Let's jump right in. So as we mentioned, Daylon and I were in Montreal for the ISSCR 2026 annual meeting, where we spent the week diving into the latest discoveries and emerging ideas in stem cell research. Beyond the incredible science, one of the highlights, of course, was getting to know and meet the researchers from around the world and hear about the work happening in their labs. We had some awesome conversations with attendees throughout the conference, and today we're excited to share a few of those with you. So without further ado, let's meet some of the researchers we had the privilege of speaking with at ISSCR 2026.
Hi, my name is Davila Arleti. I'm a director at Beam & Bio. I work on media development and differentiation kits for stem cell biology. I did my postdoc at Paul Burridge's lab at Northwestern, and one of the main projects was studying the nutritional requirements of stem cells. And with these, we developed a new basal media that accompanies the supplement that was previously developed in the lab. And we decided to branch out and make a company out of that. And we figured out that with other tweaks to this media, we can make good differentiation kits. So I do cardiac solutions on that front. That is a primary thing that was focused from early my undergrads all the way to my postdoc. And then the stem cell media. So we have a couple products that are already launched, and we're developing now other differentiation kits that might come to next year. Probably something neural lineage, hepatocyte-like cells, and other things.
Hi, my name is Carly Rodriguez, and I am a PhD student at the Jackson Laboratory through Tufts University. I study mouse embryonic stem cell models for understanding the genetics of neuronal aging and stress resiliency.
Hi, my name is Alan Giacomo, and I wear lots of hats, from advising companies about the legal aspects of information science and life science, from finance to technology. And I also teach a law school course at Hofstra that they let me design 20 years ago. It's called the Law of Medical Product Discovery, Development, and Commercialization. And this coming fall, they're going to allow me to teach a course that I just designed called Healthcare and Machine Intelligence Law.
Hi, my name is Abigail Riddler, and I'm a PhD candidate at University of Pennsylvania. So broadly, I'm interested in using the study of rare diseases to elucidate common and complex biology. So I study transcription factor GATA6, which is associated with pancreas agenesis, a rare disease where babies are born without a pancreas. But there's been recent developments with the 23andMe and such that there's actually diabetic patients who harbor GATA6 mutations and patients who don't have diabetes that have GATA6 mutations. So I'm interested in how GATA6 is specifying endocrine cells from pancreas progenitors. So I made a GATA6 Degron line in a human pluripotent stem cell line. And so I'm able to differentiate these cells, and they can have GATA6 up through the known requirements of early definitive endoderm and pancreas progenitor specification. But then I can add a small molecule DTAG to the culture media, and GATA6 is degraded within 24 hours. So it's a really clean system because up until the point where I add either DMSO or DTAG, the cells are treated the exact same way and differentiated the exact same way.
Hi, my name is Karis Johnson, and I'm a postdoc at the University of Montreal with Dr. Guy Savageau. So I am a hematopoietic stem cell biologist, and I'm currently looking at epigenetic memory after UM171 treatment in HS seeds, with a overarching aim towards optimizing these molecules and therapies for rejuvenation purposes.
Hi, my name is Laura Magnani, and I'm a last year PhD student at the Cambridge STEMCELL Institute. So we study the process of blood formation as it occurs in humans. And although a lot of things are understood about how this process occurs, some things are still unknown. And for my project, I investigated the role of the thyroid hormone, which is an hormone that is important for growth and a lot of other things in the body, but nobody has really looked into what it does on stem cells. And so my project elucidated some interesting things about it.
Hi, my name is Daniel Besser. I'm a managing director of the German Stem Cell Network, and we are also affiliated with the Berlin Institute of Health at Charité, University Medicine Berlin. So I studied biology back in the days, back in the last millennium in Berlin, even before the wall came down. Most of you will not remember that, but Berlin was two parts. So then I moved on and did my PhD in Basel at the University of Basel, and with the practical work at the Friedrich Miescher Institute. And afterwards I went to Rockefeller and went to 3 different labs there. 2004, I went back to Berlin, started a lab there. And in 2011, I decided to go more into science management. And that's how we came along with the German Stem Cell Network. There was a call from the ministry. So we got together with a couple of people, founded an association, and basically grow that GSCN. We really needed help also from the international community. That's why we always interacted with ISSCR. And we were then lucky that the Berlin Institute of Health basically from 2020 on basically gave us a permanent home so that we have funding for the rest. We have an annual meeting, which is around 500 people. We have very good interest from industry. We're trying to keep it small so that it still stays at universities. We do annual magazines. We do all kinds of things around that. We educate the public, but we also educate politicians in Germany on issues around stem cells, writing a stem cell report every 2 years, things like that. So in a nutshell, we're trying to promote stem cell research in Germany in all of its forms to the general public, politicians, but also connecting the scientists.
Hi, my name is Christian Roy-Laboy, and I'm a graduate student at the University of Cincinnati and Cincinnati Children's Medical Center. So my research mainly focuses on creating an in vitro system to understand pregnancy in the gut adaptation process, and using the organoid model system, but I have actually taken that model system and have combined it with, something that has been overlooked, which is fibroblast cells. So I'm combining fibroblasts and studying how they interact with hormones with an enteroid and organoid human-derived model.
All right, a huge thanks to everybody who took the time to speak with us for this episode. There's such a wide range of exciting research happening right now, and honestly, we're just scratching the surface. We'd love to dive deeper into each of these topics in future episodes.
Absolutely. One of the best parts of the meeting for me was getting the chance to connect with people face to face and hear about their work directly from them. Those conversations are such a big part of what makes ISSCR so special. The meeting, and honestly, it was something I'd been looking forward to for many months in the run-up Always love catching up with old friends and colleagues at the ISSCR, but more than that, I think it's about meeting new people, having discussions, broadening my scientific horizons. I, for one, had an amazing time. I can speak for Arun, I'm sure, in echoing that. We had an amazing time.
Yeah, and along with hearing about their research, we also wanted to know what had people buzzing at this year's meeting, like what kind of discoveries, sessions, themes they were most excited about. So let's hear what they had to say.
Hi, it's Davi. So one of the things that I was really excited about was seeing the applications of AI to cell biology development. So that was an interesting research. I've seen some applications of like cell therapy that are also pretty cool. Yesterday, I was talking to a bunch of CERM interns at UCSD. So that was also really interesting seeing like undergrads, first or second year undergrads doing research that is already like, oh, let's do some clinically relevant stuff. So get cells like their imaging brains to see if some CAR therapies are working for neurodegenerative diseases. So that was also a pretty cool thing.
Hi, it's Carly. Well, this is my first meeting, so we've set the bar high. I think it's been really interesting for me to learn about all that is being done with stem cells. I don't think I realized like how much it encompasses, because I mostly just think about like iPSC-derived models, and I've learned a lot about like the clinical trials that have been going on and other applications that people are using that are really interesting to learn about. Yeah.
Hi, it's Alan. The NAMS coverage. And you guys have been covering this. Every time you guys say that word, I have to stop jumping up and down when I hear that. It's just terribly important, as evidenced by all of the sessions that are concurrently running right now.
Hi, it's Abigail. I've been most excited about hearing the other trainees talk and see what cool science everybody's doing across the globe. As a patient scientist myself, I'm very excited to see all of the cool research that people are doing and how it might apply to patients and, the broader science field.
Hi, it's Karis. I think it's been a great meeting so far. Really nice representation from all sort of fields. I think the topics that have been covered are really interesting. I really enjoyed the focus on cell size and cell shape and how that seems to be integrated into our field a lot more in mechanosensing, and how physics and biophysics is becoming a more important part of stem cell biology and our understanding of stem cells, not just as a intrinsic point of view, but also as a kind of whole.
Hi, it's Laura. What I'm most excited about is all this interdisciplinarity that I see. So there are people coming from a background in nanotechnology, AI, which is a big thing nowadays, but also biology, induced pluripotent stem cells, and all these kind of fields somewhat merge and marry in this conference. So that's a high plus.
Hi, it's Daniel. We clearly now see that stem cell research has moved from basic biology, basic research. Still, it's very strong, but we see more and more things going into clinical developments, which is very important. At some point, we need to see really changes for the people, for the patients coming out of stem cell research. That's what they were hoping for the last 10, 20, 30 years, right? On the other hand, we also see very exciting things going on in drug development, disease modeling. All this organoid field has basically exploded into tons of very, very fascinating possibilities to look at drugs, possibilities to understand diseases. I think there's, if you also look at the exhibition here, at the industry exhibition, you see a lot of companies which really, make very, very important contribution into this field. So there's basically, there's tons of exciting stuff going on.
Hi, it's Christian. What I'm most excited about is the opportunity to really touch bases on things that we've overlooked. And one of the things is fibroblasts. I've seen much more talks about fibroblasts. I've even had someone come up to me and say, we used to see fibroblasts as such dull cells. And and now we're seeing actually they do play an important role, and they're there and they're interacting with stem cells, and they're doing something unique that and that's motivating and pushing the field of stem cell research.
Yeah, as you could hear from those answers, there was a lot covered at this year's meeting. Diverse research areas and speakers and some really exciting research across the board. One of the things I was most excited about was our live show with Drs. Michael May and Janet Rothberg from CCRM, who shared their expertise on scaling and manufacturing PSC-derived therapies. If you missed it, be sure to check out episode 325 to learn about the critical bottlenecks and emerging solutions that are shaping the future of cell therapy production. And then there was the schedule itself, absolutely packed, but making room for some of the networking and sidebarring and conversations in the halls. There were so many great sessions happening at once that we were constantly racing between rooms trying to catch everything. But beyond our own favorite talks, we also wanted to hear which sessions and topics really stood out to the delegates attending the meeting. So let's hear what caught their attention.
Hi, it's Davi. It was pretty cool seeing today's talk by Dr. Viv Regev from Genentech on Lab on a Loop. So the idea is you can get ginormous piece of data, do multiple iterations, and it validated some of the things that we've seen before. That is, it's not a one-time thing that you have to keep doing, iterating all the time. So it was good validation from I was an EE guy, electrical engineering guy, who developed computational models back in early 2010s. And seeing that still the length of the field that you have to keep doing these iterations, and the more data that we get, it's better. So the models become better, the predictions are better, and how to design new experiments also better.
Hi, it's Carly. There was some really interesting aging stuff. I might be a little bit biased, but looking at aging across different species, stem cells of different species, and DNA repair mechanisms and stuff. And I thought that was a really unique approach to some of these questions we have.
Hi, it's Alan. And the presenter from Genentech this morning, the last presenter, wow. She began by saying this technology, AI, has a place along the entire pathway, from discovery to development to commercialization and administration of therapeutics beyond that. And all that she was going to talk about today was that very initial step, identifying the pathway that you're going to target. You can then use that technology to design the drug or the biologic or the cell and gene therapy. But the very initial step, which takes forever, is identifying the target. So when you combine that with what can be done with respect to safety and toxicity data, you can save 1/2 to 1/3 or more of the time and expense of discovering and developing and commercializing all of these regulated medical products.
Hi, it's Abigail. I did go to a talk from a person in Christina Nostro's lab who just published a paper which was very relevant to mine, looking at a transcription factor NKX6.1 and how the level of that can help specify different endocrine populations and different beta cells. So that was a really good, interesting talk, and I was able to sit and chat with him afterwards, which was really cool.
Hi, it's Karis. So one of the plenary speakers yesterday was talking about the oocyte, and it's a subject that I don't know much about at all, but I was really fascinated to learn that the oocyte doesn't use complex 1 at all in terms of the metabolism, even though it's maybe quite expected that it would have a low energy requirement because it's a non-dividing cell. But I found it super interesting that complex 1 was completely absent from its metabolism. And she also mentioned that this was only published in 2022. So I think it's super exciting to see new fields that aren't as established coming to light in terms of women's health and studying the oocyte. And I also went to Cara McKinley's talk this morning, and it's super cool that we now have a mouse model of menstruation, and this is now a preprint, and it's kind of crazy that it's 2026, but this is where we are. So even though I'm a blood biologist and a HSC biologist, I think it's really cool to come to a meeting like this and see different fields and how these are expanding and progressing.
Hi, it's Laura. What I found really interesting was a talk during plenary session where a PI is investigating what happens to the oocytes because they remain latent for a very long period of time, and what happens during this latency in dormant state, because we know that sometimes dormant cells may accumulate, for example, in the blood system mutations, etc., but the oocytes are actually functional, so then they can come back to life and resuscitate and then give life.
Hi, it's Daniel. I mean, Yamanaka is always like very amazing. That's obviously was one of the strong talks. I heard a talk in the plenary today that was on the Drosophila intestine and related to pregnancy. I don't recall her name right now, but that was really cool stuff. I really liked it.
Hi, it's Christian. I just came from a talk about how maternal stress plays a role in fetal offsprings and the inability of the neurocrest cells to fully take over the brain development. So, it's really interesting. I've always been an advocate for maternal health studies and how that plays a role in stem cell development and embryogenesis.
Definitely some great responses there all over the map. Arun, what was your favorite session?
Yeah, I'm going to be biased and say cardioids, cardiac organoids, coming from Sasha Mengen and his team. And also Robert Zweigert, similarly, there's some amazing work happening in cardiac organoid biology and scaling these things up for not just therapeutic applications, but for development of biology, for disease modeling, for therapy, for drug discovery. So yeah, a cardiac biologist, I am biased, but there's a lot going on in the field.
Yeah, I loved Zweigert too, just because his energy, and he gave an entertaining talk, dealt with a little bit of a technical snafu with real elan and grace. You gotta love a speaker like him. I wrote down in my notes as I was watching, I would love to work in this guy's lab. Seems like so much fun. Quote. So that was a favorite for me. But truly, if I'm honest, I was looking for something this year's meeting, and I found it in Bianca Dumitrascu from Columbia University. What they say, you always find what you're looking for. And in that, I was looking for like this kind of the meeting of the modeling, the AI, the machine learning, and the biology. And I think that was a real hot topic this year. In Waxmorph, which was this new approach that Dumitrascu's lab is developing, I think it was a really vivid illustration of not necessarily what the capabilities are now, but where we're going. And I think she was very clear to fill in that there's a lot more to do with these models that are already really quite impressive. And I think there was a lot of synergy with other talks. Like I remember seeing a talk from Lim Lab that showed it kind of from a different angle, these programming with logic gates and all kinds of response elements in cells in order to model and have a kind of self-sustaining morphogenesis. So I loved this idea of the modeling and the digitization and all those digital twins. I think that was a big part of the theme this year. But honestly, there were far too many great sessions to fit into just one conversation. Every time slots seemed packed with talks we wanted to see. I couldn't get them all in. If you want to hear more about the science, the standout moments, and the sessions we attended throughout the week, be sure to check out our daily episodes highlighting our favorite moments from the meeting. We've got some more conversations with delegates in just a bit, but before we get to that,
whether you're looking to attend a conference this year or to expand your network, make the most out of your experience by downloading STEMCELL Technologies, collection of tools to help you prepare for your next event. The downloadable checklists and guides include recommendations on how to get ready before attending conferences, tips for networking, best practices for your LinkedIn profile, and more. Download the Conference Toolkit at www.stemcell.com/conference-toolkit. So earlier this year, we had the chance to catch up with Drs. Fiona Deutsch and Nozomu Yachi about what they were most looking forward to at ISSCR 2026. And one thing they both highlighted was, of course, the excitement of hosting the meeting in Montreal, a beautiful city known for its amazing food, culture, and energy. So, while obviously the science was a huge draw, we were also curious to hear about how attendees were experiencing the city itself. From exploring restaurants to sightseeing, spontaneous adventures between sessions, we asked delegates to share some of their favorite moments outside the conference center.
Hi, it's Davi. I got here a couple days before the conference started, so I was able to catch the last day of the jazz festival. And the city is like a good combination of American things, European things. Going every time between French and English is also an interesting feature. So both aspects, like the conference with interesting talks and like meeting new people, and a vibrant city, they were pretty good aspects.
Hi, it's Carly. It's my first time in Canada, and I very much love it so far. They've sold me on the city. So it's, I think, such a friendly, fun little city. I love, I didn't know a lick of French when I came here, so I love like kind of learning some more languages, but everybody's like very chill and understanding with that. And I think there's lots of different food variety options that I wasn't aware of. And I also have celiac disease, so there's a lot of gluten-free options that I've enjoyed for sure. Yeah. But I rank 10 out of 10.
Hi, it's Alan. Montreal is just such a beautiful city. I mean, it's hard to imagine a place that's cooler than Montreal. I'm sorry I missed the Jazz Festival, which just ended before we got here. I'm sorry it didn't overlap. That would have been really cool.
Hi, it's Abigail. Montreal's beautiful in general. The food has been amazing. I'm a vegetarian, so the vegetarian food has also been pretty cool, which is pretty hit or miss in the States. I really enjoyed my hike around Montreal Park one of the days before I came to the conference. It was impressive to see such a big green space and such a city environment. So it was really cool.
Hi, it's Karis. So Montreal is a really fascinating city. I don't speak French, so but it's incredibly bilingual, and it's also incredibly international. It's probably the most international city I've ever lived in, so it's been great to be exposed to a whole range of cultures, languages. You hear Spanish, French, English really commonly on the street. And yeah, it's also been great to see through the World Cup how everyone's kind of come together and how there's a lot of different pockets in Montreal of different ethnicities, and seeing them all come together and celebrate the World Cup's been really cool too.
Hi, it's Laura. I got a chance to visit Montreal before the conference. So the place that I enjoyed the most was Montreal. So literally the month within the city, because it's literally a green lung within the city. So there are a lot of people cycling, running, or just having a chill walk with a friend. So I really enjoyed walking up there, up to the cross, and then come back down. So it was beautiful.
Hi, it's Daniel. I think Montreal is a great city. I mean, it's like really amazing. I didn't had so much time to really walk around, check the things out, but I like the vibe. You know, it's very French vibe, you know, obviously. Anyhow, I think Canada is a great place, and they do a lot for stem cell research. I mean, they have a long-standing network, which we also interact a lot with. I think it's great.
Hi, it's Christian. So, loving Montreal. I think it's a very walkable city. The food scene, just like last year when I was in Hong Kong, tremendous food scene. The people are so friendly, so nice. I've been, polishing my very broken French, but they've been very, very helpful with that. And the city itself, it's much cooler weather than, down in the U.S. that we've been having a heat wave lately before coming up here. So it's been a great city and a great host. And Canada, I mean, there are our neighbors in the north, so it's really nice to interact more with them and see the cool research that they're doing up here.
Yeah, for sure. Montreal had a lot to do, and it sounded like everybody had a lot of fun. And I personally loved how the conference hall was right next to Old Montreal. this really scenic, historic part of the city with some really quaint little cafes where you can grab a bite to eat, a couple croissants here and there as well. I, of course, love the food, and that's part of the reason that I come to ISSCR every single year, especially in the foreign countries, is to figure out what's the best food to eat when we're there. And certainly that was a highlight of Hong Kong, Hamburg too, and Montreal held up just fine. So that was a big part of the visit for me.
Yeah, for me, I mean, no shade. I don't love mayonnaise, and they love mayonnaise here. The poutine was a bit of a misfire, but that's just for me. They love the poutine here. But I did love the metro. Every time I go into these meetings, I got to get on whatever the public transport is. The last few years has been a very strong metro option, and Montreal is no different. I went down to Mile End, which is a bit of a posh neighborhood, and then walked about halfway back before I got back on the metro. And what I got out of that experience was that Montreal is, I don't know about a big city, but it certainly feels big in the scope and the culture. As a few of our delegates mentioned, there's this massive green space at the center of it, the eponymous Montreal. And, you know, with that, I think there's also this old world culture, but I think close to my roots in New York City. It seemed very familiar. There was a little bit of LA in there. As I was walking by all these restaurants, they had these beautiful, straight from the farm vegetables on display. Had that farm to table feel, while there was skaters right outside under the bridge. Everything you could picture, but in a truly immaculate city, not perfectly immaculate. A bit of a misfire walking through Chinatown, grimy, at like 11 p.m. on the first night. Arun and I had a bit of a misadventure, but still, that's what you love about any one of these places is the grit, the vibrance. It feels like the pulse beating right underneath the surface, and to have a feast of science in the midst of all that, it really was a joy. So much to be grateful for at this year's meeting. Beyond the science, but at the center of it all, of course, the innovation, the imagination, and the conversations that we shared. So that brings us to the end of our ISSCR 2026 special. We had a great time at the meeting and hope that you enjoyed hearing from some of these attendees, including ourselves. Don't forget to check out all the episodes that we released during the meeting. We have that daily roundup. You can catch all of them at STEMCELLpodcast.com or wherever you get your podcasts. Until the next time, thank you guys so much for listening.